Howdy, our name is Alondra Curiel and Cassie Fernandez, and today we're going to present our lab report 12 page. The purpose of this lab is to determine the concentration of bacteriophage in a sample by performing a controlled serial dilution and plating method. Because phage particles are extremely small and numerous, direct counting is impossible, so diluting the sample stepwise allows us to reach a concentration where individual phage infections can be seen as distinct plagues on a lawn of E. coli. Each plaque represents one infectious phage particle. By mixing each solution with E. coli and embedding the mixture in molten top agar, we create an environment where the bacteria can grow while the phage infect and lyse the host cells. This technique allows us to observe plague formation and ultimately calculate the phage titer with accuracy and precision. Materials. For the cultures, we use the 10 to the first power dilution of phage, which was provided for us, and then we use the Escherichia coli broth culture. And then for the equipment and supplies, we use 9 microcentrifuge tubes, 4 LB agar tube plates, 4 large test tubes, sterile water, micropipette and sterile tips, Vortex mixer, water bath with molten top agar, 5 milliliters per sample, parafilm, and an incubator. Procedure. We started off with the serial dilution. We started off with a provided 10 to the negative or 10 to the 1 power phage tube, and then we labeled 5 microcentrifuge tubes from 10 to the 2 to 10 to the 6 power. Then we added 900 microliters of sterile water to each tube. Then we transferred 100 microliters from the 10-1 to 10-2 mix, and then we transferred 100 microliters from the 10-2 to the 10-3. Then we mixed. Then we repeated this stepwise until we reached the last one, which was 10-6, and then discarded the last 100 microliters. For preparing the plates, we labeled 4 LB plates with the date, phage type, dilution, and then these were labeled 10-3 to 10-6. And then 5E6, we added 100 microliters from each corresponding dilution from 10-3 to 10 to the 6 power into its E tube. We added 100 microliters of E. coli culture into each E tube, total of 200 microliters. Then we mixed briefly and let it sit for 10 minutes. And then for the plating of the phage, this is the part where we have to work fast. So for each plate, we poured 200 microliters of phage E. coli mix into the tube of molten top agar, which is 5 milliliters. Then we immediately poured onto the labeled LB plate. We swirled gently to coat the surface and allow the plates to solidify for 5 to 10 minutes. And then we inverted and parafilmed and incubated so we could see the results later on.
So for our results, across all the plates, we saw that the phage successfully infected and lysed the E. coli. You can see it by the all the small clearings, or also called plaques on the lawn. The plates with the lower dilutions, sort of like this one, just means it has a higher phage concentration. And it showed that they were almost fully lysed, making the plaques difficult or impossible to count. As the dilution increased, the plaques became more spread out, showing the expected decrease in phage particles. This pattern confirms that the serial dilution worked correctly and that the phage in our sample was active and capable of producing consistent plaque formation. So as you can see here, like there's a little more dispersed, but here it's like not really. And then for our T5 and T6, there's not many plaques in it, and this one almost nothing. In conclusion, overall, the serial dilution and plating method successfully allowed us to visualize bacteriophage activity through clear plaque formation on the E. coli lawn. Each dilution showed noticeable differences in plaque density, confirming that phage concentration decreases predictably with each step that these results demonstrate. With each step, these results demonstrate how dilution plating can be used to estimate phage titer and observe phage-host interactions in a controlled setting. This lab helped reinforce how precise pipetting, proper mixing, and consistent plating are essential for obtaining reliable and interpretable results. Thank you for listening to our lab report video.